My name is Jess Payne, and this is my Bio 2410 final project presentation. The first patient, the infant of Andrew and Courtney, presented with symptoms of low birth weight, misshaped head and jaw, and abnormal clenched fists. With recommendations from the pediatrician, additional medical testing was conducted and showed heart defects and other organ system malformations. To complete the diagnosis, chromosomes within the infant's white blood cells were collected and analyzed to determine if any chromosomal abnormalities were present. The resulting karyotype is shown here. After completing the karyotype, the infant was diagnosed with trisomy 18 or Edwards syndrome. This means that there are 3 copies of the 18th chromosome within the genome. Complications of this disease include mental deficits, umbilical artery aneurysm, cardiopulmonary arrest, and death from apnea, infection, or congestive heart failure. The life expectancy of children diagnosed with trisomy 18 typically does not exceed 1 year of age. However, there have been rare cases in which the child lives past 1 year. The main treatments for this disease are to treat symptoms and provide emotional support for the child's parents. Support groups are also available at the provided link. The second patient is a 5-year-old boy who presents with disproportionate growth, increased flexibility, and vision issues. To complete the diagnosis, blood was collected and an electropherogram was created to look for abnormalities in the amino acid sequences. The shown electropherogram indicates the typical sequence for a healthy individual. Within the patient's electropherogram, it was shown to have a single nucleotide mutation, the differing codon circled here. The codon within the patient was TGT, rather than the typical CGT shown. This results in a differing amino acid sequence and consequently a mutation in the protein produced. Unfortunately, this mutation causes the patient to receive a diagnosis of Marfan lipodystrophy syndrome. The disease is characterized by congenital lipodystrophy, premature birth with an accelerated linear growth disproportionate to weight gain, and progeroid appearance with distinct facial features, including proptosis, down-sounding palpable features, myopia, and normal psychomotor development. The prognosis of individuals with the disease is good, with a full life expectancy. There is no cure, so the main course of treatment is to prevent complications. Support groups are linked here as well. The last case study focuses more on research than active cases in humans. The model organism Drosophila melanogaster has proven to be successful in furthering research of the human genome and its genetic diseases. In this study, a random gene was chosen from the fly's genome and compared to that of the human genome. The gene chosen was the FMR1 gene, which is expressed in the highest amount in the central nervous system. A mutation in the homologous gene found in humans results in a disease called Fragile X syndrome. A karyotype highlighting the affected X chromosome is shown. This disease is characterized by moderate to severe mental deficits, macroorchidism, distinct facial features, and has material basis in X-linked inheritance and a loss of FMR1 function. Treatments for this disease mainly consist of symptom preventing and management. Life expectancy for individuals diagnosed with Fragile X syndrome is generally normal, with some more prone to other medical problems such as ear infections or seizures. A link to support groups for individuals diagnosed with the disease as well as parents seeking advice is provided here.